Hello girls, so we are here today to learn on chapter 6. So I hope that you will go through this video with me and make notes wherever you can. Just make the notes, and if you have any questions, anything that you don't understand, you can just leave a comment in the post below and we shall discuss further from there. Okay, shall we start now? Alright, so when we talk about cell division, we talk about production of new cells in the living organism. Why? Why do the cells need to divide? Why new cells need to be grown? For the answers, you can check the assignment that I've posted in previous posts, okay, asking you why do cells need to be produced. I think you have found your answers there. If you haven't, go back to the assignment, check it out, and then we shall discuss it further. Alright? Okay, so before we begin, in this slide you can see that there's a structure of a cell. Okay, this cell structure and organization you have learned in chapter 2, correct? So in this chapter, you are going to recall back a few of the organelles or the structures that are involved in this cell division as well. So firstly, you will see that the cell membrane, cytoplasm, nuclear membrane, nucleus. Okay, in the nucleus we have the chromosomes. So if you forget what chromosomes are, you can go back to chapter 4, all right, and then you can see there what are chromosomes made up of. Okay, the nucleic acid, the DNA, the chromatin, and so on. If you remember, then you can proceed. If you don't, you go back to chapter 4, check on it, then you come back and watch this video. Okay, so if you have checked again on chapter 4, let's recap a little bit before we go in further, just in case you don't understand. Alright, so chromosomes, what are chromosomes made up of? Chromosomes are made up of supercoiled chromatin. As you can see here, this is the chromosome, how the chromosome looks like, and they are made up of the supercoiled chromatins. Okay, the chromatins are the thread-like structure, okay, formed from DNA molecule and also with histone. Okay, and histone and this chromatin will form a nucleosome. All right, and then all these supercoil, and then it is form chromosomes. All right, so when you see this structure, okay, this is the chromosomes. All right. Now, chromosomes are thread-like structures found in the nucleus of the cell, and chromosomes are made up of DNA. Now, a chromosome, all right, it has chromatin. So when you draw, let's say I have this is a centromere. Never mind, it's supposed to be circle. This is a centromere, and it binds to chromatin. Okay, we call it the sister chromatin that binds together and forms chromosome. Okay, so this is the centromere and this is the chromatin. Okay, both of these will be known as sister chromatin. One chromatin and this is the sister chromatin bound or bind together at the centromere. Okay, if you don't understand, draw it down and label them, then you move on to the next video. Okay, next is we are going to see two vocabulary that you need to know. Okay, karyokinesis and also cytokinesis. Karyokinesis is the nuclear division and cytokinesis is cytoplasmic division. Division means nucleus divide or cytoplasm divides. Okay, so there are two, karyokinesis and cytokinesis. kinesis, nucleus division and cytoplasmic division. This is the chromosome, like I've explained. Structure of chromosome looks like this. Okay, and sexual reproducing organisms, okay, have chromosome in them. For example, chromosomes are found in somatic cells and also reproductive cells, or we call it the gametes. Somatic cells are every other cells in the body that divides, that has nucleus, that has DNA. Okay, and reproductive cells are the one that undergoes meiosis process, whereas the somatic cells undergo mitosis. So mitosis and meiosis are the process that you are going to learn in this chapter. But before we go into these processes, there are certain things that you have to be cleared first. Okay. Now there are two types of cells in the body: the non-reproductive or the somatic cell, like I have explained in my previous slide. Okay, so we have, for example, the phagocytes, the neurons, and so on. All right, and then the gametes are the reproductive cells in the body or in plants. For example, the pollen grains and also the spermatozoa, sperms, eggs, all the reproductive cells in animals and in plants. So this chromosome in somatic cells are diploid in number, okay, 2n, and the cells the chromosomes in the gametes are haploid in number, n. So that means the gametes have half the number of chromosomes compared to the somatic cells. Why? We will see further in this video. So this is just a brief introduction, okay, on mitosis. Look at mitosis. Alright, mitosis, when it is carried out, it produces two cells, okay, two cells that are identical to the parent cell. For example, my skin layers divides, okay, the new cells of the skin form, and it is the same as my previous cell, as the parent cells. But meiosis are different. Meiosis only involves with the gametes. That means there are from the sperm and from the ovum. So here they must have half a number in sperm, half a number in the egg, so that when it is fertilized, so the zygote will have the exact number as the parent cell. Okay, so don't worry about it. We will see in meiosis process 6.3. But for now, you just know that meiosis produces haploid number of chromosome and mitosis produces diploid number of chromosome. Meiosis produces 4 cells, 4 daughter cells, and mitosis produces 2 daughter cells. Okay. Alright, so human beings have 46 number of chromosomes. That is why human beings are said to have diploid number of cells. Okay, so each chromosome consists of a DNA and a protein. So diploid cells are said to have 2 sets of chromosomes. 2 sets of chromosomes means 46 chromosomes in total. And haploid cells said to have 1 set of chromosomes, which is 23 chromosomes in total. So now look at this picture here. This one. What can you see here? How many sets of chromosomes you have here? There are 23 sets, 22 plus X and Y. X and Y are the one that determines your gender, male or female. So we have 23 sets here. But if you count, we have 46 chromosomes. 46 chromosomes meaning this is a diploid cell. Okay, diploid cell. Now, the human genome, okay, genome means the total number of chromosomes in human is 23 homologous chromosomal pairs. So we have 1, 2, 3 until 23 pairs of homologous chromosomes. So these are the sex chromosomes, 23 set of chromosomes, which total up equals to 46 number of chromosomes, not set. This is 23 set, 46 chromosomes. So number of chromosomes are different in all organisms. Not all have 46 set of chromosomes. For example, in the ant there are only 2 chromosomes. In fruit flies they have 8, carrots have 20, dog have 48, shrimp has 254, and so on. So you can see in this slide, not all animals have the same number of chromosomes, but human beings have 46 chromosomes. Okay, now we will go through cell cycle and mitosis in the next subtopic. So you know about cell division, you have recap a little bit on chromosomes, number of chromosomes, you know what is diploid, you know what is haploid cells. So now we are going to look at cell cycle and now the cell cycle is a sequence of events that involves DNA multiplication and cell division to produce 2 daughter cells. Okay, cell cycle, okay, cell division to produce 2 daughter cells, mitosis. So in the cell cycle, we have 2 phases, which is the interphase and the mitotic phase, M phase we call it. So in the interphase, we have G1, S and G2 phase. We will see what is these phases all about. So basically the cycle is on growth and reproduction of new cells. Now the main stages in a cell cycle, okay, you can see here the first one is mitosis, which is a nuclear division, karyokinesis, and we have the cytokinesis, which is the cytoplasm division, and then we have the interphase, okay, which is the growth phase that happens 90% of the time. Okay, and this interphase is the one that has the G1, G2 and the DNA synthesis phase, the S phase. Okay, so this interphase, now we'll go through interphase. Okay, G1 is gap 1, S phase is the DNA synthesis phase, and G2 is the gap 2 phase. Okay, so this is the cell cycle. Okay, I hope you will draw this diagram in your notes. Okay, and take note of each phase. So the first growth phase, that means the G1, gap 1 phase, is the growth and normal metabolic roles happens here. Okay, this is all under the interphase. And then we have the S phase, which is the DNA replication, that means synthesizing phase. It happens here, DNA synthesizing, replication, and so on. And then we go into the second growth phase. Okay, that means the G2, where the growth and preparation for mitosis happens. Then it goes into the mitotic phase, which happens to be the mitosis process. Okay, and mitosis process, we have here 4 levels of phases. That means we have the prophase, metaphase, anaphase, and telophase. All these are under mitotic phase. Then after the mitosis, we have the cytokinesis. Okay, I will repeat. Look at G1 phase. Okay, G1 is where the growth and normal metabolic phase, growth phase, number 1. Then we have the S phase, which is the DNA replication, and then the G2 phase, where the growth happens together with preparation for mitosis. And then comes in, this is number 4 already, number 4, mitotic phase, where it happens. And then after that, we have supposed to have here is the cytokinesis process phase, cytokinesis phase. That means the cytoplasm divides and then 2 cells are formed. Okay, so now mitosis. What is mitosis? Mitosis is the process of nuclear division which results in the formation of 2 genetically identical daughter nucleus. So meaning that the nucleus divides to form 2 daughter cells, and the number of chromosomes of the daughter cells are the same as the parent cell. So this whole process is known as, okay, mitosis occurs in animals and in plants. Okay, in animals, it is in a non-reproductive or somatic cells, and in plants, it is in the meristem cells or the tip of root or a shoot. Okay, so this also explained just now what is somatic cells. So both animals and plant cells have mitosis. Now let's do the vocabulary check before we go into the phases of mitosis. Okay, now centromeres, centrioles, and centrosomes. What is centromere? Centromere is the point of constriction of the chromosome. Look at here. Okay, this is the chromosome. Okay, there are two sister chromatids here that are bind through the centromere. So this is the centromere that binds the sister chromatids. Then we have centrioles. Remember centrioles? Okay, it's only found in animal cells, not in plant cells. Okay, and they occur in pairs. That means they come in pairs. There are two pairs in each cell. Okay, and they lie at right angles to each other, and they are able to self-replicate. So mitotic center, you can see here, these are the centrioles, and they will form spindle fiber. We will see what are spindle fiber all about. Okay. Then we have centrosomes. Centrosomes are the organelles located near the nucleus. They are not membrane bound and they serve as mitotic center in animals, but they are absent in plants. They divide and migrate to opposite poles of the cell during mitosis. So we have here are the centrosomes. These are the centrosomes here. Okay. So the centrosomes are not centrioles, are responsible to assemble the spindle fibers, and the spindle fibers are formed during mitosis and both meiosis. It arranges the chromosome into their correct positions in preparation for cell division. So it's actually holding the chromosomes together. These are the spindle fiber, and there are the centrosomes. Okay, spindle fiber, the spindle serves as the railroad track along which the chromosome will move, and it's a framework that keeping the two poles apart. Alright, so you can see here the chromosomes that are attached to the spindle fiber. Okay, how they are aligned together. So the spindle fiber are the one that helps the chromosome to move. Okay, next we'll go on to interphase, a stage prior to mitosis. That means before mitosis, interphase happens. The G1, the G2, and the S phase. Okay, both in animal and okay. So here, prior to mitosis, how does the cell prepare before it carried out mitosis? Okay, that means each chromosome will now exist as pair of chromatid. Okay, they are joined together by a centromere. Don't understand? Draw it down. So interphase is a stage where each cells are 4n. There are 4 copies of each DNA molecule, 2 in each chromosome of a homologous pair. Okay, so this is a homologous pair. Okay, and these are the copies of each DNA molecule. So we have, this is the homologous pair. This is the homologous pair, 2 sets of chromosomes. So now, why are chromosomes drawn in different color? When you draw chromosomes later, you will draw blue and red, because to differentiate the chromosomes from maternal and paternal origin. So it's okay. You can also do shading. One you shade as black and the other white. Now let's look at the mitosis. Okay, so the stages in mitosis in the cell cycle is the prophase, metaphase, anaphase, and telophase, correct? So PMAT, how do you remember this? Okay, we can remember as Professor Mutt. Professor, prophase, Mutt, M is metaphase, A anaphase, T telophase. Professor Mutt. Now, in mitosis, the parent cell, will undergo DNA replication. The chromosomes are copied and then it will double in numbers. Then the chromosomes will split. They will split and divide. And the daughter cells are said to be identical to the origin of the parent cells, but it has the same number of chromosomes with the parent. In prophase is usually the longest phase of division. The chromosomes shorten and thicken by coiling. Condensation happen. Condensation means chromosomes shorten and thicken, and the individual chromosomes can be seen. It's no more in chromatin form. It's no more coil. So each chromosomes can be seen. This is a picture of late prophase, where the nucleus and the nucleolus have disintegrated. This is a real cell. And in prophase, the nucleolus will disappear, the nuclear membrane will break up, and the spindle fiber is formed. You can see the picture over here, and you can draw later on the phases that I'm going to describe here. After prophase is metaphase. So when the chromosomes are now formed, condensation happens. These chromosomes will now line up around the equator. This is the equator here. There's a line in the middle of the cell. Equator of the spindle, and attached to their centromere to the spindle fiber. So each centromere is attached to the spindle fiber. You see here, they are all attached. They are all attached to the centromere. And then the metaphase plate is an imaginary plane through the center of the cell where chromosomes align. Let's imagine the plate here. And the chromosomes align at the metaphase plate. And the centromeres are attached to the spindle fiber. Next comes in anaphase. This phase is very rapid, very fast. The centromeres will quickly split into two. One chromosome, and then it snaps. It snaps in between here. Alright, and then the chromosomes will move to the opposite pole. This is pole number 1, this is pole number 2. So you can see the centromeres are now split. This is the centromere, and the sister chromatids are also split. So the spindle fiber will pull the daughter centromeres to the opposite poles. The separated chromatids are pulled behind the centromeres. Now, why are the chromosomes in V shape? As the chromosomes are pulled towards the pole, at the centromere, so it will form the V shape because it's being pulled by the centromere. So now comes in the telophase. The telophase is where the chromosome reaches the poles of the cell. Just now anaphase is just dividing, and now it has reached the opposite poles, waiting to divide, waiting for the cytoplasm to divide. So in this phase, the chromosomes are uncoiled and lengthened again. It's like the reverse of prophase. Right? Spindle fiber will then disintegrate, and then nuclear envelope will reform, nuclearly reappear. It's just like a prophase, but now the cells still haven't divided yet. And mitosis is now completed at the end of telophase. Then comes in cytokinesis. Cytokinesis after telophase, whereby the cell now will divide into two. The cytoplasm will divide. So cytokinesis happens after telophase. So after that, after telophase, after cytokinesis, two identical daughter cells are formed. That means they are having the same number of chromosomes as the parent cells. Okay. Okay, now we are going to see the difference between mitosis and cytokinesis in animal cell and plant cell. Now, cell division is still not complete at the end of mitosis. As the division of the cytoplasm has not yet begun, cytokinesis must happen after mitosis. So, cytokinesis normally follows telophase and then leads into the G1 phase or back into the interphase. Okay, so you can see here, okay, this is the interphase, and then preparation for mitosis happens here, and then mitosis process happen, the prophase, metaphase, okay, anaphase, and then here comes telophase, here telophase, and this is cytokinesis, where cytokinesis now takes place. That means the division of cytoplasm. So cytokinesis in animal cells. This is how the cell looks like. Okay. So when it divides, when the cytoplasm starts to divide, this is known as a cleavage furrow, where it will divide and form two different cells. So this one you can see how it looks from the electron microscope. In plant cell, they don't have the cytoplasm dividing without the cell plate. So the plant cell will form the cell plate in between the cells, and then it divides. The cytoplasm. Okay, so the difference between mitosis in plant versus animals. So in plants, there are no centrioles and centrosomes, but in animal cells, there are centriole and centrosomes, where spindle fibers and all are involved in dividing the cells. And in plant cells, cytokinesis happens by formation of cell plate, whereas in animal cells, cytokinesis happens with the help of cleavage furrow, where the cells divide into two. Now, the necessity of mitosis. Okay, for this you can refer to your textbook, page 104. So, mitosis are very important for various processes, life processes. For example, the first one, for embryo development with organism and organism growth. So, mitosis ensure that rapid cell growth occurs from baby until you are an adult and so on. The cells will keep growing and growing. Alright, and then through mitosis process, the lizard is able to grow a new tail if the tail breaks, regeneration process. Okay, next is when the body is injured, mitosis will produce new cells to replace cells that are dead or damaged. Okay, and mitosis also aids organisms such as Hydra to produce new individual through the formation of new buds. Okay, so we can see mitosis is helping all the somatic cells to. In plant cells, in agriculture, the mitosis process, okay, the technique of culturing plant tissue is used to produce young plants through the culturing of parent cells without going through the fertilization process. Okay, this culturing technique uses the stem cells from animals, which are then cultured in laboratories to produce meat. And then stem cell therapy uses stem cells from bone marrows to treat damaged cartilage. So there are many functions, necessity of mitosis in our life processes. Okay, the end. So we are done with 6.1 and 6.2. If you don't understand any of this video, you have to let me know by commenting in the post below, or you can also share some extra information if you do your own research, you find something online or from your textbook that you don't understand, get to me, we shall discuss in further and make sure all your classmates know about it. Okay, so after this video there will be an activity for you to do. Okay, so you have to refer to the post and do the activity given, and you are going to draw each cell in all the four processes, the prophase, metaphase, anaphase, and telophase, that occurs in mitosis. So you're going to do that. Look at the assignment given and make sure you do your notes. Don't forget, for 6.3 we will come back in another video. Okay, goodbye.